Okay, good morning. I'm Dave Dorman, and I'd like to welcome everyone to the peer review meeting for the draft NTP research report on the Clarity Bisphenol A course study. And what I'm going to do is ask everyone at the table to introduce themselves. I'll start off with myself. I'm David Dorman. I'm a professor of toxicology at the College of Veterinary Medicine, just down the road at North Carolina State University. Nigel?
Nigel Walker, Deputy Division Director for Research for the NTP here at NIEHS.
Barry Delclose, I'm from NCTR, Division of Biochemical Toxicology, and I was the PI on the study.
Louisa Camacho, NCTR, Division of Biochemical Toxicology, and I was the primary co-PI of the study.
Greg Olson, NCTR pathologist.
Paul Felton, NCTR statistician.
Tracy Bunton, I have a private toxicology consultancy for drug development. I'm also a veterinary pathologist.
Hi, Tom Rosal. I'm a veterinary pathologist, and I'm the chairperson of biomedical sciences at the medical school in Athens, Ohio, at Ohio University.
And I'm Gonçalo Gamboa da Costa. I'm the project officer on the FDA side to the interagency agreement under which this work is conducted, and I'm also the liaison officer to the National Toxicology Program.
I'm Janet Tews. I'm a professor of biostatistics at Wake Forest School of Medicine.
I'm Kim Trinan. I head up a toxicology group at a pharmaceutical company, Synovian. I've been in the business on pharmaceuticals and looking at regulatory reports for about, 20 plus years.
Mike Connor, pathologist, toxicologist, currently with Global Blood Therapeutics, is heading up their non-clinical safety department. And yeah, I've been in the pharmaceutical business for about 30 years and academia for about 10 years before that, so. Sorry.
I'm Mary Wolf, NIEHS NTP, and I'm the designated federal official for this peer review.
Good morning. I'm Bill Slicker, Director of the National Center for Toxicological Research. Welcome to everybody.
I'm Brian Barrage. I'm the Associate Director of the National Toxicology Program.
We can also go ahead and begin introductions of other individuals in the audience, starting at my left, please. So maybe...
This is John Bucher, NTP.
Cynthia Hines, National Institute for Occupational Safety and Health.
Dave Malarkey, the NTP Pathology Group.
Retha Nebo, NTP.
Heather Pattisall, North Carolina State University.
Scott Belcher, North Carolina State University.
Ernie Hood, Bridport Services.
Atlee Watson, NTP.
Troy Hubbard, NTP.
Keith Shockley, NIEHS.
Amy Bricks, contractor for NTP.
Georgia Roberts, NTP.
Suramya Vaidyanatha, NTP, NIEHS.
Michelle Huth, NIEHS, NTP.
Vicki Sutherland, NTP, NIEHS.
Kristen Ryan, NTP, NIEHS.
Steve Henches, American Chemistry Council.
Kelly Shipkowski, ICF.
Candon Bird, ICF. Jeannie Liu, ICF.
Ginger Guidry, Office of Communications, NIEHS.
Ken Fuji, Coal Safety Research Institute.
Thad Shug, Division of Extramural Research and Training, NIEHS.
Okay, thank you. Norm, are you on the phone? You won't be able to. So on the phone is also Dr. Norman Barlow, who is our liaison back to the NTP Board of Scientific Counselors. So welcome, Norm. Hopefully you can hear us. And I'd like to take a moment to reintroduce Dr. Barrage, who's currently the new Associate Director of NTP, and for some opening remarks. Brian?
Thanks, David. So as David said, this is my first opportunity to sit in this chair, so I appreciate the opportunity to be a part of this. I have been witness to some previous peer reviews, so I recognize the importance of this process, and it is an important part of our ensuring that the data that we put out in these studies has won the full force of our collective learnings and perspectives, but also has the impact that we hope that it will have on policymakers, regulators, and the general public. Just a few contextual things. I think we all recognize that you know, a lot of this work that we do, despite our best intents, is not perfect, right? We design the best studies we can. We make the best interpretations and observations. We painstakingly review those things to make sure that the quality is what we expect it to be. Again, this peer review process is an important part of that, to challenge perspectives, and I've tried to get up to snuff as best as I can, even though I'm coming sort of late to this game, and I've so reviewed some of the initial comments from the peer reviewers, and so I think this will be some really good discussion. I think there's good challenge in here, and I think it will be a learning opportunity for all of us. At the end of the day, I think we all recognize biology's hard, and pathobiology is even harder, and so there is probably no right answer. If there's anybody here who knows all the right answers, I really would love to meet you. So what our intent will be, again, to put our collective learnings perspectives together and ultimately come up with as good an interpretation as we possibly can so that it has meaningful impact for the folks that we consider the stakeholders. This particular study is rather unique in that it is a sort of a guideline study, the core study, but it has lots of other elements. And so as all those elements are put together, we'll have some different perspectives that will emerge. So this peer review process is really about peer reviewing and challenging, debating, considering the outcomes of the core study. So just as a reminder, there's lots of additional data that will be integrated with this core study. So with that, I want to reinforce David's welcome to everyone, and in particular appreciation to our peer reviewers, because it is not an inconsequential effort to critically evaluate these reports, this data, and offer perspective. So thank you for that.
Okay, thank you. I'll turn the mic over to Mary Wolf now, who's the designated federal official. Dr. Wolf.
I'll read the conflict of interest statement. The members of the NTP peer review panel for this research report serve as individual scientists and not as a representative of any organization. Each member is to exercise judgment prior to the meeting as to whether a potential conflict of interest might be present due to his or her occupational affiliation, professional activity, or financial interest. This potential for a conflict also applies to the member's spouse, minor child, partner, or organization. Panels are asked to bring to the attention of the federal official any potential conflict, and it will be handled in accordance with departmental policy and regulatory requirements. I'm going to pass around the post-meeting or the meeting conflict of interest just for you to attest that your conflict of interest status has not changed since we initially did your review. If you'd please sign. I'll ask everyone in the audience to please sign in at the registration table today so that we have a record of who's currently in the room. There are people watching the webcast, but we do not collect their names unless for the minutes. Breaks, lunch are at the discretion of the chair. For those of you in the room, there is wireless access. It's NIH Guest Network. For the panel, whenever we start the peer review and you give your comments, if you'd please state your name and please use the microphones. After the peer review, we will be preparing a draft peer review report that will be shared with each panelist so that you can review it and ensure that we correctly captured your comments. You will not add any new information, but we want to be sure that you are agree with how we captured your remarks at the meeting. For the audience, the slides from this meeting will be posted to the NTP meeting website. It'll take about a week because we have to make them compliant for according to federal guidelines for accessibility. We're also capturing and going to archive the video from this meeting today, and it will be posted to the NTP meeting website in about a month. Again, it will take that much time to get the captioning synced with the video and make it compliant for federal accessibility requirements. At the end of the day, the panel will be taken to the airport or back to the hotel, whichever is appropriate for you. We'll have a shuttle. Thank you very much.
Okay, thank you. I always love, at the discretion of the chair, we will take breaks. I'm going to be trying to keep track of the schedule as close as possible to what was discussed with Dr. Wolf and others, but I'm old enough, I need breaks, so we won't be going through all day with nonstop. So what I do want to do is spend a couple of minutes just talking about the overall format of today's meeting. So we have a pretty full agenda, and it's going to be important for us to stay on task, both as presenters as well as panelists. One thing that I will say at the end of the day is, you know, our job is to peer review. Our job is not to rewrite the report. And so we'll want to refrain from attempting to wordsmith things at the very end of the day. So what we'll be doing is we'll basically have a series of presentations from the PIs. I don't know why we're getting all kinds of feedback. We'll get presentations from NTP and NCTR staff regarding the CLARITY study itself, the major findings and interpretations. We as a panel will have opportunities to ask clarifying questions at each break. And then we'll end up having identification of those written public comments, and we'll have a slide showing you who's provided written comments. And then we do have two individuals that will be giving us oral public comments about mid-morning or so. And then each reviewer in advance of today's meeting was assigned different tasks. And so then what will happen is during the formal peer review portion, each reviewer will present their comments. Just also keep in mind, as Dr. Wolf explained, and we'll talk about it again, although you've got pre-written comments, you're not obligated to cover every single one, and what you say in the public record today is actually what gets said. And so just keep that in mind, that the pre-comments that you provided in written draft form, those are not part of the formal peer review unless you enter them in deliberately today. And then there'll be opportunities for us to have more interactions with staff to discuss and clarify our comments. And then at the end of the day, there's a series of interpretations that NTP has developed, and what we'll be doing is voting on those either collectively or individually, and it kind of depends upon how the peer review process goes this afternoon. Our goal is to be done about 4 o'clock, and we'll go from there. I do want to acknowledge that there's been a lot of effort on the part of the public to provide the committee with written comments. I think there were quite a few very insightful comments that may have helped focus the attention of the peer reviewers, and I do appreciate that. And again, as far as the oral comments are concerned, although we only have 2 individuals, we'll still stick with our plan of having 5 minutes per speaker and then 1 speaker per organization. There will be opportunities for the members of the panel to ask some clarifying questions, but again, we're not trying to get into a prolonged debate with the oral comments, okay? So that's basically what's ahead of us, right? Okay, so what I'm gonna do is check my watch, because I will do that periodically with my really fancy high-tech phone here, and we're actually 4.5 minutes ahead of schedule. So I'm going to turn it over to Dr. Walker to give us an overview of the study.
To kick things off is actually give you an overview of the whole program. We're going to have Louisa is going to give a detail on the core study details itself, but my role here is really to give you the background, the genesis of this whole CLARITY program, where the core study fits into the larger program, and then we'll have an opportunity for comments. So essentially, I'll break it down over the next 20 minutes, 4 areas: background, where we came to, how it came to this, the core study outline, and then some of the program management and the next steps on this whole CLARITY program. So many of you know bisphenol A is widely used in the making polycarbonate plastics and epoxy resins. This has been a long-standing concern of the public about the widespread exposure to bisphenol A, which is generally pretty low in the less than sub microgram per kilogram per day, from migration from small amounts into foods, from water bottles, cans, those kind of things, very low level exposures. But there's been, over the last 10, 20 years, considerable debate over what is the actual risk posed by that low level exposure. Some of the early guideline studies that were conducted on bisphenol A at higher concentrations didn't really show anything. But there's been a lot of investigative studies since that time, many of which have been funded by the National Institute of Environmental Health Sciences, showing, reporting that BPA induce a wide variety of effects in various model systems, exposures much closer to those of human exposure. So that actually is creates a bit of a regulatory issue for the Food and Drug Administration. The current regulatory landscape for FDA is that it maintains that BPA is currently safe at the current levels occurring in foods. FDA also supports the current use of BPA in food containers and packaging. And this comes from a continuing assessment of the science that's available on BPA from both past guideline studies, academic studies. There was a major reevaluation risk assessment of BPA back in the late 2000s, and then subsequent reevaluations of the information as it comes out on top of that, reviewing the current scientific evidence to come to that particular conclusion. From an NTP perspective, we've been involved in BPA for quite a while. Notably, back in the mid-2000s, the Center for the Evaluation of Risk to Human Reproduction, CRHR, that subsequently became the Office of Health Assessment and Translation, did a monograph review of the available scientific literature at the time about the possible effects of BPA on human development and reproduction, specifically in that area. This came out in late 2008 and the main conclusions were on the 5-point scale that CRHR had at the time, was there was some concern for developmental toxicity for fetuses, infants, and children, particularly with respect to effects on the brain, the behavior, and the prostate gland. There was lower levels of concern for other areas such as the mammary gland and early puberty, and higher exposures in reproductive toxicity in workers. And so this really set the stage for what NIEHS and NTP might do next, in that this information was also provided to the FDA at the time, and they had concluded around then that the NOEL that was appropriate for dietary intake was about 5,000 microgram per kilogram per day. This provides an adequate margin of safety at the estimated levels at the time. But they also noted when they did that review that there was insufficient evidence and information to support that other information could be used as a point of departure for potentially developmental prostate gland toxicology, developmental, neural effects, or behavioral. And the FDA Science Board that reviewed that assessment by FDA recommended further research to address some of these underlying concerns and questions. And actually, some of the specific recommendations were that a rodent study should be considered, specifically designed to meet normal regulatory guideline acceptance by FDA for evaluating compounds, should address some of these endocrine-related questions that have been raised, and use additional endpoints and models to actually assess these endocrine-mediated developmental processes. They also encourage the development of physiologically based pharmacokinetic models to help better understand the translation of rodent and non-human primate pharmacokinetics to the human experience, particularly with regards to developmental exposures. And I'm not going to go into any of that work, but a huge amount of work was done at NCTR in that respect by Dan Dorge's group. And so that will also comes into the mix of how we integrate all this information. So specifically for CLARITY-BPA, the inception of this, what CLARITY stands for is the Consortium Linking Academic and Regulatory Insights on BPA Toxicity. That's a bit of a mouthful, so we generally just call it CLARITY, because hopefully we're going to provide some clarity to some of these questions. It was, I think, I see Thad is in the audience, and I think Thad and Jerry Heindel, who in extramural at the time were key members of the team putting this together, actually came up with an acronym, and it's been, thankfully we did, it was good. And really it was the development of this idea that we're going to try and address some of the uncertainties about BPA toxicity, use a relevant long-term oral dosing protocol that includes developmental exposures to interrogate, and this is the key, additional endpoints that you would not normally interrogate in a traditional guideline toxicity study. And that would actually use a core program, a core exposure paradigm conducted under GLP that will allow everything integrated into the same exposure system. This was really a novel collaborative approach that neither NIEHS or NTP had really engaged in. Quite the idea of bringing a lot of folks together. And so essentially it takes a village, and this took folks from the extramural community in the NIEHS, staff from the NTP, the folks at NCTR and CIFSAN over the past about 8 years now, of actually putting and managing this program together. And the key part of this is we were going to use NIEHS funded grantees to add those additional endpoints that you wouldn't normally see in a guideline, interrogate in a guideline study. And the way that came about was back in 2010, there was a specific funding opportunity announcement. This was to develop this consortium of researchers that could work with NTP and NCTR to address some of these, hypothesis-driven mechanistic studies to add information to addressing some of those uncertainties that have been identified in 2008 and the FDA Science Board. So these proposals were solicited. The applicants went through the traditional NIH scientific peer review for selection. Also, the projects were then subsequently subjected to a technical evaluation to see whether they could actually be done within the context of integrating within the guideline study. And finally, in 2012, everyone kind of got together down at NCTR and hammered out the final design of that. And that's when we came up with the consortium name of CLARITY. And so basically this program has been going on now for about the last 6 years. And so it's quite satisfying to see us come to this quite a major milestone within the development of this program. The key components, as Brian alluded to, is the core study, which is the subject of this particular peer review meeting. This is a 2-year chronic study conducted at FDA, NCTR, and Good Laboratory Practices. And it was essentially designed in accordance with classic guidelines for conducting chronic toxicity and carcinogenicity studies. Also, the key component is the grantee studies. There were 14 academic investigators that were pulled into to add information and studies to this particular project, focused on a range of different endpoints, and both molecular, structural, functional. And the key point is they use siblings from the core animals exposed under the same conditions as the study that we're reviewing today. So this really enhances the ability to integrate across all the different types of studies, both the grantee and the core studies, to look at what was the totality of the effect of BPA in this particular model. And that is going to comprise the third aspect, which will be the integration report that will occur after all the core study data is available and all the grantee study data is available. We're going to be pulling all that information together. That project is going to be led by John Bucher here at NTP, and to see what was the totality of the effects. From a cartoon point of view, essentially this is animals were exposed to either vehicle, BPA at 5 different doses, or a reference estrogen. And again, there was both continuous and stop-arm exposures. And then from those, animals went to either the core study or to the grantee studies. And so what you can see is a very integrated approach here to look not only at the effects of BPA, to put it in context of estrogen and estrogen control, ethinyl estradiol, because there was a lot of questions about whether BPAs and estrogen, and particularly at low doses. So to get into the actual core study, the details, I'm going to start layering on information, and Louise is going to get into even more detail. The model choice was the NCTR Sprague-Dawley rat. This is was maintained as a breeding colony at NCTR, originally established in the 70s from the Charles River Sprague. And the strength of having this model is all the breeding has been controlled over the years, and so the interaction between sibling matings and not getting any drift, that was a key part of this, was using a well-established line. The other thing about the NCTR colonies is they tend to have lower background tumor rates than we've seen with some of our other NTP strains that we've used in other classic NTP models. We know that this is responsive to estrogens because this was the model that was used for NTP's endocrine disruptor initiative in the mid-2000s, where we were looking at a variety of estrogenic agents, particularly ethinyl estradiol, genistein, and nolphenol, looking at the long-term and transgenerational impacts of exposure to estrogens. We also know that it's responsive to BPA. Barry did Dr. Delclos did a study of BPA before the CLARITY program to show that this is responsive at high doses to BPA. That data was already published. And therefore, we also know that this strain can be used for we understood the pharmacokinetics also in this strain. So we've got an integrated set of data showing that it's both BPA responsive, estrogen responsive. We understand the pharmacokinetics in quite detail. And so that was the beauty of using this particular core model. The dose rationale was very different than you would normally see in a traditional 2-year cancer bioassay, because this was a dose response assessment study. This wasn't about hazard ID at the high doses that would normally be driven by doing a 90-day study, identifying minimally toxic dose, and then going from there maybe 2- or 4-fold dose increments down. This was really to cover the wide range of doses with reported effects in the literature. So in this case, it goes from 2.5 to 25,000 microgram per kilogram per day. 2.5 is only 10-fold higher than the absolute lowest limit that we could get from removing all potential routes of exposure in the diet to BPA. So it really is almost the lowest feasible exposure we could have done in this particular model. And the top dose, we felt provided a sufficient margin of exposure. We're looking at almost 25,000 X margin. And again, the focus here was on the dose response, not necessarily just the high dose effects. The reference control, as I pointed out, this was a question that had been in the literature of whether BPA is an estrogen at low doses. And so the inclusion of ethinyl estradiol was added to the study. The doses were informed basically based on not only our endocrine disruptor studies, but also on the 90-day study that Barry did prior to the CLARITY, and those have been all reported before. So this actually, the size of this study actually creates some challenges for interpretations for the panel today and for us when we were going through it. There are actually 56 exposure groups. Just to put that into, because you've got 5 doses, control, stop exposure, continuous exposure, 1-year interims, 2-year interims, males and females, and 2 estrogen reference control groups. That's actually 7 times the size of a normal rat arm of a traditional cancer bioassay that we would do. So that's going to be both a statistical and an interpretive challenge because we're seeing a lot more groups than we would normally see and interpret from in a study. The other challenge for the interpretations is the 10,000x dose response range. Many of you have probably seen GLP chronic studies, and they are generally in the 4x to 10x dose range. We take a high dose, we maybe go half logs or 2x or 4x down. In this case, we've got a 10,000-fold dose range. So that challenges the expectations of what you might expect of a dose-dependent response. Because normally a traditional NTP bioassay would fit in one change from the dose groups. And again, the third challenge is the historical experience. We actually, while this has been used for many studies, those were done back in the mid-2000s. There are very few recent long-term studies in this particular strain, and that again. So that is a challenge of having the appropriate historical control for this particular concurrent exposures. And the other historical experience is NTP has never done an in vivo study with this number of groups, this size, in a study longer than 90 days. So this type of large, very wide dose response range with so many groups, we've never had to deal with this before. So again, we're bringing our experience from having done lots of other bioassays and long-term chronic studies and tox studies, but we've never had something exactly like this. So again, that's a challenge to how we interpret. I'd like to give you, so that's the basis for the core study. I'm just going to leave you now with some how the whole program was managed and some of the next steps on the wider CLARITY program. So the program was managed over the last 6 to 7 years by a steering committee that include representatives from NTP, NIHS, NCTR, CIFSAN, the Regulatory Center at FDA, and the researchers from the grantee institutes. We had regular conference calls, regular webinars coming together on progress on the study. We also had an external scientific panel that gave us advice from time to time. There was Martin Filbert, Dan Cassiano, and I'm blanking on the third person. Strange. Mike Gallo. I can't remember Mike. Okay. I'm good to see, actually, recognize some folks around the room here. I see Jerry Heindel and Thad Shug. They were the extramural representatives on the steering committee over the years. John Bucher and myself were the program officers for the NIHS NTP aspects. And Gonzalo is also now the project officer of the interagency agreement. Rita was one of the advisors on the team, Rita Newbold. And so there's a lot of, it's really taken a village over the last 6 years of folks coming together just on managing this program. And a key part of this is actually also not only the management of the program, but management of the data. This produced a huge amount of animals, a huge amount of information, and the NCTR went to amazing lengths to control for everything, to manage all the animals, where animals were going. We had 14 different researchers around the country getting animals at different times. That was a logistical challenge. I think Louisa might get into some of the challenges that posed, because it was a very large study. The data all was collected blinded for the grantee studies, and all that information has now been deposited in the NTP's Chemical Effects in Biological Systems database before it was actually decoded and sent back to the grantees. So some of the concerns in use of academic research is, that's you don't know what wasn't collected. Well, we collected everything from these studies, and the grantees were very engaged in wanting to do this blinded. We have that information all in the databases. It's currently still blinded to us at the NTP, but all that information is going to be released later this year. This is the 14 list of the 14 grantees that are actually providing additional information, as Brian pointed out, to this large project, and they're covering, as you can see, a wide variety of different health endpoints, from heart effects to the brain to neurobehavior to the mammary, prostate, all those areas that were of concern in the original SEER monograph back in 2008. The grantees have been trying to address some of those endpoints using additional approaches that you wouldn't normally include in the guideline study. And so in terms of the program's next steps, so we have here today, we have the NTP research report for the core study. That is the draft being reviewed today. We anticipate the final report after addressing comments and looking at all the peer review comments and public comments. We anticipate that to be completed probably by in the summer. August is hopefully our time frame for this, to have that publicly out. Grantee publications have been ongoing during this program. Different grantees had samples at different periods during this whole project, and so have been publishing and are continuing to publish their own research on this. The data sets I have pointed out are all in SEBS, but what we've wanted to do is to make sure that those can be released to the public in a common format, so that when people come, it's not so discrepant people can't understand it. And so our SEBS team here at NTP has been working closely with the grantees to put this into a common format, so that people can come, download, and actually integrate data themselves between the different data sets. So that includes not only the grantee studies, but also the core study data. These includes methods also on how the data was collected, and we're anticipating all that data will be ready for public release by the end of August as well. And then, as I pointed out before, the integration report will be occurring after that has all occurred. And we anticipate that's going to occur through 2018, through into 2019. And so watch this space for another peer review on the whole CLARITY program sometime, I guess, in 2019. The data is currently from the core study is all available, and we created an exclusive CLARITY-BPA website, where you can get the core study now, and this will be where the grantee data will be available later in the summer. There's the website. And with that, I'll take any clarifying questions, and then I'll take a seat, and if you have any more questions, thank you.
Okay, thank you. Any questions from the panel?
Tracy Bunton.
Hey, Tracy.
So when you say the grantee data will be integrated with each other, and then is that integrated with the core report saying, okay, this is the stop dose group and et cetera?
Yeah, so different grantees got different samples. I didn't go into a lot of detail. Some at PND 5 and 21 and 90 and 1 year. Most of that data was collected up through the 1 year time period. And so the idea is to bring all that data together and integrate and see whether effects occurring at earlier times and lower doses using potentially more sensitive endpoints that we can then match up with the effects in essentially siblings and sibling groups and exposure groups. So it might be both an interpretive integration where what do we see here versus here. There may actually be some actual mathematical analyses trying to bring different statistical approaches. That plan has yet to be fully fleshed out at this point.
Okay, thank you.
Okay, thank you.
Turn it back to Dr. Wolf.
Put the charge up. So can you please put the charge slide? So just to remind the panel and the audience that the charge to the panel is to review and evaluate the scientific and technical elements of the CLARITY-BPA core study and its interpretation, and its presentation, and to comment on NTP's interpretations of the data. Does anyone have any questions?
Moving right along. In case you're wondering, we will have extra time for the meat and potatoes portion of the presentations later. So, right?
Yeah, that was just the.
So, next speaker is going to be Dr. Luisa Camacho from NCTR. The floor is yours.
Thank you. And I will apologize already. I'm suffering a lot from allergies, and North Carolina is not helping. So if I have to cough at some point, I apologize already. So over the next, I think the slides are not advancing. Okay, now it's working. Thank you. So over the next 30 minutes or so, I'll go over the study aim, give you a bit more detailed overview of the study design, and also some discussion on the exposure of the study animals to the BPA background exposure. So the aim of the CLARITY core chronic study that we'll be discussing today was to characterize the toxicological potential of bisphenol A or BPA following a perinatal only or a chronic exposure in both male and female rats exposed to a wide range of BPA levels. As was mentioned before, the animal model we used was the NCTR Sprague Dolly rat. The reasoning for that was our extensive experience in characterization of the potential toxicity of a series of estrogenic compounds, including E2, nonylphenol, and genistein. And this animal model was also used in our previous 90-day subchronic study on BPA. And in addition, we have quite comprehensive knowledge of the pharmacokinetics of BPA in this animal model, and so we will be able to better interpret any toxicity outcomes in relation to the internal dosimetry of BPA. So in our study, we took extreme care to try to avoid, or in cases where we could not avoid, to try to minimize and monitor exposure of the study animals to both BPA and other estrogenic compounds. So potential sources of BPA in animal rooms include material made of polycarbonate plastics, since BPA is the monomer for these plastics, And so in our study, we did not use polycarbonate cages and water bottles. Another source of BPA is feed. So our previous study, which has been published by now, we assessed the quantity of BPA that could leach from several diets that are commonly used in toxicity studies, including 5K96, NIH41, 501, AIN93, and all these diets had low but detectable levels of BPA, so in the range of a few parts per billion. And so these data suggest that actually all animals, if the animal eats, it will be exposed to a certain amount of BPA. So the most we can do is to try to monitor for it and establish maximum acceptable levels of BPA in feed, which we did for this study. Animals, so feed is also a potential source of estrogenic compounds such as phytoestrogens. So especially if we use soy-based diets, it can have up to hundreds of parts per million of genistein and daidzein, so we avoided using this diet in our studies. And also bedding can be a source of mycoestrogens if using corn husks bedding contaminated with fusarium. So we avoided all these exposures by using special animal housing conditions. And so this included the use of polysulfone cages, glass water bottles with silicone stoppers and metal sip tubes. We used milliport filtered water that was provided ad libitum, and we used hardwood chip bedding. In older animals that developed pododermatitis or ventral masses, at the recommendation of the Veterinary Services for Animal Welfare, we transferred some animals to alpha-dry cellulose bedding, which is a softer bedding. And all these transitions were recorded. So all these housing materials, we checked them for leaching levels of BPA. So both old and new cages were checked. Glass water bottles were checked periodically over the study. And all lots of the hardwood chip bedding and of the cellulose bedding were confirmed to have no leaching levels of BPA before being used in the animal rooms. As I mentioned, we used a special diet that was not based on soy, that did not contain soy and alfalfa. It was a low phytoestrogen diet. And again, we have extensive experience with this diet in combination with this animal study in chronic and multi-generation studies that we've conducted before at NCTR. All feeds of diet were tested before being provided to the animals for the dietary components, for contaminants, phytoestrogens, the mycoestrogens relanone, and also BPA. And we had established the maximum level of BPA, acceptable level of BPA in the feed to be 5 parts per billion. And so based on estimated feed consumption, this would afford exposure to BPA through the feed that was about 10-fold lower than the lowest dose of BPA being tested in the study. So due to the size of the study, we had to use 11 feedlots. And I'm here just listing the average levels of the genistein, daidzein, cholesterol, and zearalenone, and also of BPA. And as you can see, the mean value of BPA in the feed was 1.28 parts per billion, which afforded about 1 to 8% of dietary exposure to BPA, lowest than the lowest dose. Sorry, 1 to 8% of dietary BPA exposure compared to the lowest dose of BPA being tested in the study. So the test articles, the primary one was, of course, BPA. The BPA solid was air milled to create a finer solid, and this helped ensure the homogeneity of the dosing suspensions of BPA. Before being shipped to NCTR, the test article was thoroughly analyzed by Battelle, and upon arrival at NCTR and also over the progress of the study at the end of the study, both the identity and the purity of the test article were certified. In addition to BPA, we had another test article, ethinyl estradiol. As was mentioned before, we use this as a reference estrogen control. And again, the identity and the purity of this test article was certified at NCTR prior to the study start and periodically over the study and at the end of the study. So we had 8 dose groups in this study. We had a negative vehicle control. We used 0.3% carboxymethyl cellulose, again, to help in the having an homogeneous dose suspensions for the higher doses of BPA. We had 5 BPA dose groups ranging from 2.5 to 25,000 micrograms per kilo body weight per day. And as was mentioned before, the rationale for this range of doses was 2.5 would afford at least a 10-fold higher margin of exposure to the maximum allowed dietary intake of BPA, while the 25,000 dose was considered to afford a sufficient margin of exposure compared to the current estimated human exposure, which is estimated to be less than 1 microgram per kilo body weight per day. And in addition, we had 2 doses of the reference estrogen control E2 set at 0.05 and 0.5 micrograms per kilo body weight per day. And these were set based on our previous data from the 90-day subchronic BPA study. The dosing formulations were certified to be within 10% of the target concentrations. They were used within a pre-established window of stability. And in the case of the highest doses of BPA that were suspensions, we also ensured that they were homogeneous, so they had plus or minus 10% of homogeneity. And in addition, also we verified periodically over the course of the study the dosing vials, meaning that we were going to the animal rooms, get some of the dosing vials, analyze them, and make sure that the label matched the contents. And this was verified for all doses. The animals were dosed by oral gavage 7 days per week. We used 5 mils per kilo body weight per day of dose volume, and this volume was calculated based on the daily or weekly body weight. So daily body weight that was collected just prior to dosing in animals younger than PND 90, and or weekly body weight in animals in the continuous dose that were exposed after PND 90. The gavage volume was dispensed automatically using a Hamilton pump. These gavage pumps were certified to deliver within 10% of the target dose prior to being used and periodically over the course of the study. We had 4 dosing stations per animal room, and the doses were administered from low to high dose in each station to avoid cross-contamination between doses. Also, the pumps were thoroughly flushed between dosing formulations and were cleaned at the end of the day with a previously certified washing method. So, as I mentioned, we had 4 pumps, gavage pumps per room. The first pump was used only to dose the vehicle control. The second pump was used to dose the 3 lowest doses of BPA. The third pump was used to dose the 2 top doses of BPA, and the fourth dose pump was exclusively dosed to the reference estrogen control E2. And here in this photo, I'm just showing you the dosing setup. So we had the computer that was connected to the gavage pump and to a balance. So the animal will be, the body weight of the animal will be taken at balance, recorded by the computer, which automatically would calculate the volume of gavage to be dispensed, and that information would be transferred to the gavage pump, and the animal could be dosed. So both body weights and the volume of the gavage were recorded in our system. In terms of exposure, So the F0 pregnant dams were exposed daily from gestation day 6 until the start of parturition. And once the litters were born, the pups were directly dosed from postnatal day 1, so from the day after birth. All pups were dosed from PND1 until weaning at PND21, at which stage some of the pups were allocated to a continuous dose arm, and so in that arm, the daily gavage proceeded until sacrifice at 1 or 2 years of age. The pups allocated to the stop dose arm, they did not receive any further daily gavages after weaning, so they were just kept and then again sacrificed at 1 or 2 years of age. So the continuous dose arm just follows a standard guideline exposure for chronic toxicity studies that include the perinatal exposure. So it's the one probably most related to the continuous human exposure. A second arm was included in our study, the stop dose arm, in which, as I mentioned, the animals were exposed only during the perinatal period. This dose arm was included due to the interest in assessing potential effects of hormonally active compounds upon perinatal exposure only. As I mentioned, the animals were not gavaged after weaning at PND 21, so the two arms are not really directly comparable. Basically, just due to the size of the study and the limitations on the logistics, we could not include stop dose arm for the EE2. But we did include a continuous dose arm for EE2. Another element of design that is unique to our study was that rather than relying upon lactational transfer of BPA from the dam to the pups, we actually directly dosed the F1 pups after birth. And this decision was taken, again, to better mimic human exposure during the perinatal period, and also because our previous pharmacokinetic studies indicated that the transfer of BPA through the milk is very poor. So if we had used gavage the dam rather than the pup directly, probably the exposure during the perinatal period would be very minimal. So due to the size of the study, we had to use 5 matings or loads. These were spaced 4 weeks apart, and each load included 120 breeding pairs. And this number was selected based on past reproductive performance from our studies. And so this included taking into consideration both the success of mating and also of littering. And I will say that we're exactly spot on. So we had planned for 78% success of mating and littering, and that was exactly what we got. So we did not allow matings between litter mates or first cousins, and the breeders were transferred from the NCTR breeding colony to study conditions, that meaning polysulfone cages, the special diet, glass water bottles, at PND 21, so when they were weaned, and they were mated only at weeks when they were so they were weaned at 3 weeks of age, and they were mated around 10 to 15 weeks of age. And the allocation period occurred between September and December 2012. So the breeders were paired, so 1 male to 1 female, and allowed to mate for up to 10 days. Every day of the breeding period, the females were checked for the presence of a vaginal plug, or if that was not detected, a collection of a vaginal smear. So a vaginal smear were collected. And when a vaginal plug was detected or a vaginal smear was positive for sperm, that was considered evidence of mating and considered gestational day 0. And if there was no evidence of mating after 10 days, so the breeders were just removed from the study. So for the F0 females, they were removed from the study if they did not produce a litter by gestation day 20. This was probably indicative that we had missed the true mating day. If they did not deliver a litter by gestation day 26, or if they produced a litter that had insufficient size. And the dams were also removed from the study upon weaning of their pups at PND 21. So in the case of these last three types of dams, they were all sent to pathology to assess uterine implantation sites. And although this presentation is supposed to be only on study design, but I'm already saying that there were no differences in the number of uterine implantation sites among those groups for any of these types of females. The F1 litters were not allocated to the chronic study if they originated from litters with insufficient sample size, which again is less than 3 pups per sex. They were randomly culled to a maximum of 5 males and 5 females on the day after birth, so postnatal day 21. And as I was mentioned before, they were weaned at postnatal day 21. And up to 3 males and 3 females per litter were allocated to the chronic study, with the stipulation that no same-sex litter mates were assigned to the same dose group within the same dosing arm and sacrifice time. And this was stipulated so that the litter was the unit of analysis for the postnatal after weaning endpoints. So upon allocation of all pups, at the 1 year interim sacrifice, continuous dose arm, the vehicle, control, all BPA doses, and E2, they had between 20 and 26 animals per sex per dose group allocated. And in the case of the stop dose, again, we didn't have the E2 dose group, so only vehicle and BPA, we had between 19 and 22 animals allocated per sex per dose group. And for the 2-year terminal sacrifice, again, for the continuous dose group, vehicle and BPA, we had between 46 to 50 pups allocated per sex per dose group. In the case of the E2, our target was 26, so we fulfilled that target. And in the stop dose 2-year terminal sacrifice for the vehicle and BPA, we had 46 to 50 animals allocated per sex per dose group. I will mention that for the 1-year study, our original target was 26 pups per sex per dose group, which we did fill in, but we were falling a bit short on some of the CLARITY-BPA grantee studies. And so a decision was taken among the CLARITY consortium members to transfer some animals from our chronic study to grantee studies. And so that's why the target here is a little bit lower than 26. Just to give you an idea on how the animals were housed, so we had 2 breeding rooms in which 1 male and 1 female breeders were housed per cage. Once evidence of mating was detected, the pregnant female was transferred to what we call the gestation room, where she was started dosing at GD6 until she delivered the litter. Once we had a litter, the litter and the dam were transferred to a 3rd room, the pre-weaning room, where the pups were started dosing at postnatal 1 until they weaned. At this point, up to 3 males per sex per litter were assigned to our post-weaning rooms, to the chronic study, where 2 males or 2 females, 2 females or 2 males per cage were housed, and the remaining siblings were allocated to the CLARITY-BPA grantee studies. And in each room, we had representation of all dose groups, all dosing arms, and sacrifice times. The in-life endpoints included morbidity and mortality, checked twice daily, clinical observations, the dam body weight that was collected daily prior to dosing, Once the pups were born, we counted the number of pups alive and dead on the day of birth and on the following day, so postnatal day 1. At postnatal day 1, we also determined the sex ratio and the weight of the litter per sex. And after culling, we started collecting individual pup body weight, which was used to calculate the volume of gavage. The individual body weight was collected through PND21 in the case of the stop dose arm, or through PND90 in the case of the continuous dose arm. And in all cases, the body weight was collected weekly after that. We collected a series of vaginal and vaginal cytology-related endpoints, which I'll explain further in the next slide. Palpation of the F1 animal started weekly after 6 months of age. And we also collected feed consumption data, mainly to be able to estimate the background dietary intake of BPA throughout the study. So in terms of vaginal cytology related endpoints, these were collected from 26 randomly selected females per dose group per dosing arm that had been allocated to the terminal sacrifice. Starting on PND 22, we start monitoring for vaginal opening. And then at about 16 weeks of age, we collected 14 consecutive days of vaginal smears to be able to assess estrocycle lengths. And after this, we collected 5 consecutive days of smears every month to be able to assess estrocyclicity and reproductive senescence. And this monitoring was stopped when the animals were considered to be in aberrant cycling, which was defined as 2 consecutive months of 3 consecutive days of estrus or 5 consecutive days without a day of estrus. And the first months of this aberrant cycling was considered the time of aberrant cycling. In terms of necropsy endpoints, so for the 1-year interim sacrifice, we assessed hematology, clinical chemistry, sperm parameters, which included testicular spermatid head counts and cauda sperm counts, motility and morphology. We also assessed body and organ weights and did histopathology on 21 selected tissues, which are listed here, as well as any gross lesions that were found at necropsy. And due to the interest on dorsolateral prostate, so we did assess 6 step sections that were cut at 100 micrometers intervals through the dorsal prostate. All the other tissues were processed following standard NTP specifications. In the 2-year terminal sacrifice, we assessed body weight and histopathology similar to what was done in the interim animals. So the histopathological evaluation was performed by the study pathologist, who also saw the necropsy and was responsible for writing the pathology report. His findings were evaluated independently by a quality assessment group, and the pathology data review was conducted by QA pathologists. And in the case of this study, we did 100% review of slides of tissues of particular interest, which included ovary, the uterus, vagina, testes, epididymis, prostate, seminal vesicle, male and female pituitary, and male and female mammary glands. And just to give you an idea of the size of the study, so only the review of these pathology data accounted to over 19,000 slides, which is quite amazing. Then these data, the NCTR data, was reviewed independently by a pathology working group who evaluated potentially treatment-related lesions. also looked into disagreements in diagnosis between the study pathologists and the QA pathologists, and also at rare spontaneous lesions of general interest to pathologists. And so only after all the histopathology data was peer-reviewed, it was considered final, and then it was sent for statistical analysis. So for the statistical analysis, the data were analyzed within sex, so males and females were analyzed separately, within dosing arm, within sacrifice time, and also within test article, meaning the BPA dose groups were compared to vehicle independently of the E2 dose groups compared to vehicle. For all endpoints, liter was considered the unit of analysis, and the NTP standard statistical procedures were followed, with the exception of some additional tests which we included in this study to look at histopathological data, and I'll give more details in the next slide. The tests applied to the non-histopathology data were two-sided and were corrected for multiple comparisons, the only exception being the trends for abnormal astrocycle, which were one-sided. In the case of the histopathology data, and this is normal at the NTP studies, these data were just, the tests were one-sided, and they were not corrected for multiple comparisons. And for all endpoints, the statistical significance was set at 0.05 level. So the tests applied for the histopathology data, what we called in the report the primary statistical tests, were the commonly used NTP tests, which look at incidence of lesions only. And these tests include, for the interim sacrifice, where survival was not an issue, we used the Cochrane-Armitage test for trends and the Fisher's exact test for the pairwise comparisons. And we call the combination of these tests the CAFE test. And as I mentioned, these tests are not adjusted for survival. For the terminal sacrifice, where survival was an issue as expected, we used the POLIS-3 method, which is adjusted for survival. And I should point out that all these tests, they do not assume monotonicity across dose levels, so they will be able to detect potential low dose effects. Because a lot of the non-neoplastic lesions, they have, our pathologists also attributed a severity score. So we conducted additional statistical tests, which we call the secondary statistical tests, which take into account not only the incidence but also the severity of the lesions. These tests are not adjusted for survival, and they included the Jokier-Terpstra test for trend analysis and the Shirley-Williams test for pairwise comparisons. Because the Shirley-Williams test assumes monotonicity of doses, so it will not be able to detect a potential low dose effect. So we conducted yet an additional test called the relative treatment effect test that again takes into account both lesion incidence and severity, but does not assume monotonicity across those levels. Okay, that was the overview of the study design. So the last few minutes, I just want to give some background, some information about the potential for exposure of study animals to background BPA and some discussion on this issue. So as I mentioned before, from the get-go, we knew that all study animals, including the vehicle control, would be exposed to certain levels of BPA. Because of this, we monitored for BPA in the feed being the known source of BPA in the animal rooms, and we established a priori maximum level of BPA allowed in the feed of 5 parts per billion. So we quantified BPA in all feedlots, and the average was 1.25 parts per billion. That ranged from below the limit of quantification for that day up to 3 parts per billion. So all lots were within our maximum allowed dose level. And so based on this mean dietary exposure, and also because we calculated the feed consumption, so we were able to have a pretty good understanding of what was the intake of BPA by the study animals. And so this was estimated to to range between 0.03 to 0.2 micrograms per kilo body weight per day. And so this corresponds to 1 to 8% of the lowest BPA dose being assessed in the study, which was 2.5 micrograms per kilo body weight per day. So in addition to this dietary exposure, we are also presuming that there may have been exposure of the study animals to additional low levels of BPA. This exposure would have happened through an unknown source. We could never really locate it. And it's presumed to have happened during the co-housing of load 1 animals from the chronic study in the same room as the load 0 grantee animals that were being dosed with a dose 10 times higher than the chronic BPA dose levels. And so this dose was 250 thousand micrograms per kilo body weight per day of BPA. I should point out that the time where the load 1 animals and load 0 animals were co-housed was during the gestation and pre-weaning rooms, since after weaning, the CLARITY-BPA grantee animals and the core study animals were housed in different rooms. The reason we are assuming that this cross-contamination may have happened is based on our previous data from the 90-day subchronic BPA study. And these data have been published by Churchill et al. So in the 90-day study, we had included BPA measurements in the serum to characterize the internal dosimetry to BPA across life stages. And because of that, actually, we also realized that there was an intended exposure of the study animals to BPA. So in this 90-day study, the highest dose used was 300,000 micrograms per kilo body weight per day, so in the range of the highest dose in the load 0. And what we came to realize was that the serum levels of BPA glucuronides, which is a metabolite of BPA produced only in vivo, so the levels of BPAG were similar in the naive and vehicle controls, as well as in the lowest dose of BPA, which again was 2.5 micrograms per kilo body weight per day. And so there was some contamination to low levels of BPA of all study animals. So in the case of the chronic study for CLARITY, we could not test if this unintentional exposure also happened. The reason being that, first of all, we had not planned a priori blood collection for internal dosimetry assessments, since this had been already characterized in the 90-day study, and the CLARITY-BPA study was already pretty complex as it was. And by the time that we had collected and analyzed the BPAG data from the 90-day subchronic study, all load 0 animals, so the co-housing between load 1 animals and load 0 animals had already ceased, since this was performed right in the beginning of the CLARITY-BPA study. So we didn't have any more animals to collect blood from. However, we did collect blood from the chronic study animals 1 week prior to the interim sacrifice and assessed the BPAG levels in their serum. And in these assays, we represented animals from all loads and post-weaning animal rooms, and the blood was collected at approximately Cmax for the continuous dose arm, which was 15 to 60 minutes after the last dose, or at a similar time of the day in the stop dose arm, since these animals were not being dosed anymore. And what these data shows is that in these CLARITY-BPA animal rooms, where the top dose was 25,000 micrograms per kilo body weight, we could not detect BPAG in the vehicle serum, suggesting there was no unintentional source of BPA. As I mentioned, when the animals, Clarity animals, were co-housed with the load 0 animals, in which the top dose was 250 micrograms per kilo body weight per day, we could not determine if the serum levels of BPAG, but due to the similarities with the 90-day study, we are assuming that there may have been some unintentional background exposure of the animals to levels of about 2.5 micrograms per kilo body weight per day. And so this unintentional exposure would have affected the load 1 animals, which represent about 20 to 25% of the animals in each dose group. So to try to address these from a statistical endpoint, we conducted sensitivity analyses. These were aimed to determine the impact of potential unintended exposure of study animals to BPA on the statistical analysis outcomes. And so what we did in this sensitivity analysis was we conducted the tests as described before, but excluding all animals that had been co-housed with load 0 animals for any period of time. And we could do this because we track each animal over time, so we knew exactly in which rooms they were housed in any particular time point. So in the report, if the effects found in the sensitivity analysis were significant, that were not significant in the inclusive analysis using all animals, we noted them in the reports. The reverse we did not note in the reports, meaning if an effect was significant in the inclusive analysis but not in sensitivity analysis, we did not make note of it. As an overall conclusion, our findings show that both the inclusive and sensitivity analysis outcomes, statistical outcomes, were very similar. And so these would indicate that the potential unintentional exposure of the study animals to the low doses of BPA did not impact the outcome of the study. And with this, I will thank you for your attention and take any questions.
Okay, thank you. Are there any questions from the panel? Yes?
You said that litter was the unit of analysis, but then the animals for most of the experiment were assigned at weaning. So within litter and sex, they were assigned at weaning to different dosing groups. So I'm confused how litter could be the unit of analysis.
Right. So because we analyzed the males and females separately, and so we only used one male or one female per liter. So in essence, liter was the unit of analysis. So we only had one representative per liter in a particular dose group within sacrifice time and the dosing time, the dosing arm. It is.
But if I'm understanding your question correctly, though, I thought that your question probably relates to the F0 to the dams as opposed to the F1 generation. Because it was the dams were allocated at weaning as opposed to, correct? As opposed, because the F1s were dosed at PND1.
Right, well, but they were exposed gestationally through the dam. So we are considering the liter, meaning the dam, as the unit of analysis.
Right.
Since all the pups that
Right.
were born from that liter, they are
Right.
related in essence.
Right.
Mm-hmm.
I just have a question about the dosing and the rinsing between doses. I assume that you assessed that if there was carryover, and what were the dosing apparatuses flushed with?
So yes, so prior to the study start, we tried different washing procedures, and we used the highest dose being used on that pump, and then did washing. So first we used water, which was not enough to get all especially the high dose BPA, which was a suspension. So what we ended up doing was 1 minute wash with water, followed by 1 minute flush with ethanol, and then 1 minute wash with water, and that got rid of all the carryover.
Thank you.
So just a quick request for the panel members, if you could please identify yourself prior to you doing the question. That way, as the records are being collected here for what we're saying, they can be attributed to each one of us. So just as a quick reminder of that. So I guess I'll have the prerogative too of asking maybe a question or two. So you did a lot of effort trying to look at, for this individual study, BPA exposures, but then do you have any indication whatsoever of what historical BPA levels might have been and the impact it would have on your historical controls?
So we assume, so of course we don't know, we didn't look for BPA in feed over times because that was not in our radar. But based on all the, and you know, historically probably polycarbonate cages were used because those are cheaper and they are more resistant. So, but honestly, the cages, the drinking water bottles made of polycarbonate, they reach very low levels of BPA. So the main source of BPA in the animal rooms really is from the feed. Although we cannot go back to look at old lots of diet, but the levels were pretty consistent across, you know, the 11 lots that we used in this study and also other types of diet we quantified BPA on, and they were all in the very low parts per billion levels of BPA. So I'm assuming the exposure would be similar, or if not lower, because we did not have the very high doses of BPA being used in the animal rooms, which seems to be the source of contamination.
Just Nigel Walker. And also the 5K96 was used in those endocrine disruptor initiative studies in the 2000s, right, Barry?
That's
so
correct.
It's the same type of feed, just wasn't analyzed.
Mm-hmm.
Other questions? Sure.
Tracy Button, and this is also for later, but I thought maybe you could answer it now. You had an interim sacrifice group, and I can understand some of the reasons why you did that, because you're going to get better data on some of the evaluations. But could you just go through all of the rationale for having that group?
The interim sacrifice?
Yes.
Yeah, so we could assess additional endpoints, as you mentioned. So we could assess clinical chemistry, hematology, organ weight, which at 2-year sacrifice, those data are not so useful anymore because the animals have aged by then. So it's easier to have, it's better, we get better data if you do an earlier sacrifice. And also in terms of the histopathology, we could assess progression of lesions.
That's what I thought, so thanks.
Go ahead.
Yeah, I'll add another clarifying to that. Also, several of the grantees were looking at more chronic endpoints, and so there was also the opportunity that in order to get grantee animals for a 1-year time point, we needed core study animals. And so it kind of is a double whammy in some ways that we can look at progression, early time points, and also provide chronically exposed animals that have been looked at in grantee studies all the way through the 1 year. Particularly, I think some of the groups were looking at the mammary and the prostate for that.
Tracy Button again. So were there other peel-off groups for the grantee institutions within the study, or did they only get animals at the same time points?
No. So actually their animals were separate. So we first allocated pups per liter to our study. We gave it priority. And then the leftover, I mean not leftover, but the other siblings on the litter, they were allocated to the different grantee endpoints. So for the grantees, actually we had several different time points. We provided animals at postnatal day 1, 15 for the thyroid endpoints, PND 21, PND 90, 6 months and 12 months. So 12 months was the longest exposure period for the grantees. And these different time points, they were based on the, on the study design and rationale for each of the grantees. So not all of them got tissues over these different time points. It was really driven by their hypotheses and target organ.
Okay, so seeing no other additional questions, I want to thank you for your presentation. And we are actually 1 minute ahead, so we will stay on schedule. We are going to take a break, and we'll reconvene at 10:15. And so our next presentation is by Dr. Delclos from NCTR, and he's going to present actually a series of 3 shorter presentations regarding the study findings from the CLARITY core study. Floor is yours.
Okay, thank you. And what I'm going to try to do is present in some coherent fashion a summary of the results of study and then the preliminary conclusions that were presented in the draft report. And I know that Louisa just did a thorough job of describing the protocol in great detail, but before I started discussing the data, I wanted to do a brief summary again of the main features of the study and how we were going to look at the data. So the F0 generation were brought on to study conditions at weaning and then raised until they were adults without treatment. There were very minimal data. The body weight data were collected near the time of assignment to dose groups and was used for weight ranking, but that was it. There was no analysis done. There's then mating period done, and these dams were dosed starting on gestation day 6. And then at the time of parturition, was stopped dosing. The pups were dosed from PND1. On the litters, the dams gestational and litter parameters were collected, and that was it. On PND1, treatment of the animals started. We had 2 arms, the continuous dose arm, where the animal was then dosed throughout the entire study, and a stop dose arm down below, where the animals were already dosed to PND1, and they were taken off any dosing and maintained on diet until the interim 1 year or the terminal or 2 year, and I'll be using those terms, interim, terminal, meaning 1 and 2 year sacrifices. The vaginal opening and vaginal cytology data were collected on both sets of females. Males and females were analyzed separately, and the dose arms were analyzed separately. At the 1 year interim sacrifice, we collected the organ weight, clinical chemistry, hematology, sperm evaluations, and histopathology. At the 2 year time sacrifice, we conducted only the histopathology investigations. So I'm basically going to go through down the list, similar to the way it was organized in the technical report, the endpoints that we looked at, from survival, gestation, litter parameters, body weights, the vaginal opening and cytology data, the other data collected at 1 year, and then the histopathology, which will be the bulk. I'm not going to spend a lot of time on all of these endpoints. So first we're going to talk about the survival and early removals. We monitored survival several periods during the study, the pre-weaning period and the period between weaning and the interim and the terminal sacrifices. In summary, and for BPA, there were no statistically significant effects in any phase of the study on survival. EE2, there was a limited statistically significant effect in the low dose in the pre-weaning animals. And then the moribund removals and early deaths increased substantially after 1 year of age. So this is just a summary on this slide of the percent survival in the pre-weaning animals, and then the interim continuous dose, and at the bottom stop dose animals. And as I mentioned in the last slide, there were no statistically significant effects on survival except this 1 point here at the low dose EE2 in the females in the pre-weaning period. The survival was good throughout the pre-weaning period of above 90%. The data was good at the interim sacrifice times for both the females and males, and the continuous and stop dose animals, with the male control being the lowest, 82% for this time period. The removals and early deaths, this is just an example slide. I'm not going to show you all the survival curves. This is an example from the continuous dose females, where, again, after 1 year, the early removals increased, and most of these, showing on the next slide. Okay, so the continuous dose at the top, female and male, stop dose at the bottom, female and male, and the survivals ranged from around 20% or 17% up to 40%. They were similar in both the continuous and stop dose. The majority of the animals were removed as moribund, and therefore we were able to get a complete histopathological examination of the tissues, and including a cause of death evaluation on the early removals. The most common causes of removal or death between 1 and 2 years were for the females, mammary gland fibroadenoma, and I'll show those numbers later on. Pituitary adenoma carcinoma, mammary gland adenocarcinoma. In the males, the pituitary adenomas and carcinomas, followed by nephropathy, perpustral gland carcinomas, and malignant lymphoma, spleen, were major causes of death or causes of removal. Gestation and litter parameters. I'll be very quick on this because all I've done is listed the endpoints. There were no significant treatment effects of other BPA doses or EE2 on the implantation sites, the gestational body weight gain, litter size, litter sex ratio, the number of pups born dead, litter weight, or I've already discussed the survival, which was the other endpoint measured on the litters. Body weights, overall summary. We monitored body weights, as Louisa described, at various intervals across the study. The males, there were no statistically significant effects of BPA or EE2. For the females, there were statistically significant effects. There were few, and they were confined to restricted time periods in the intermediate dose groups, and I'm showing those instances on the next slides. This is the pre-weaning body weight in the females, and you can see at these early time points there's a statistically significant lowering at the low dose of EE2, approximately 5%, between 5 and 10%. We considered that a minimal effect of not worthy of note, too much note. This is in the post-weaning body weights from the interim animals, and there were no statistically significant BPA or EE2 effects in either sex or study arm. We did make note, and this is not a statistically significant effect, but we had noted in the 1-year animals, and the 1-year animals and the 2-year animals were analyzed separately, that the low dose BPA group was higher. I don't know if you can see on the slide, there's a 95% confidence interval drawn, but it's above the 95% confidence interval. So we wanted to take a closer look at that. It was about 10 to 13% higher than controls from these weeks 36 to 52. In the next slide, I'm showing the post-weaning body weights in the interim and terminal to 1-year animals for the vehicle and the low-dose BPA. And what you can see is that the highest here is the curve that I showed in the last slide. If we had actually done these, pooling them together, there would not have been a significant effect. There wasn't a significant effect, but it would not have been a notable increase. So that does not appear to be of interest. The other significant effect, in the terminal animals, for the males, there was no significant effect of BPA or EE2. For the females, there was a statistically significant effect in these 3 highest doses, weeks 96 to 104 at the end of the study, that were higher than the control in the 250 BPA dose group. The others did not reach statistical significance. As we go out here, we're having a decreased N and a higher variability in these older animals. So they were the body weight effects observed in the study. I'll briefly cover the data that was collected on the vaginal opening and the vaginal cytology. This is a repeat of the slide that Louisa had shown, but I wanted to make a reminder before we started. We took 26 animals per dose group and monitored them for vaginal opening. And then from these same animals at 16 weeks of age, vaginal smears were taken for 14 consecutive days to evaluate estrous cycle. And then from these same animals, for 5 consecutive days, vaginal smears were taken monthly. And Louisa had described the method that we used to determine an animal was going into aberrant cycles and removed. We realized going in, this is a crude method, This is a method that we had used years ago when we did multi-generation studies with chronic studies with genistein and phenylestradiol. And we had discussed before going into the study how could we improve the method. It doesn't give us a good window of when the animal actually is going into aberrant cycles. It gives us only a wide window. But we couldn't come up with a practical means. We could not possibly do smears on all these animals every day. So the data are what we had to settle for. So neither BPA nor EE2 affected the timing of vaginal opening at the doses that we tested. And I will show in subsequent slides that the high dose of EE2 had a very clear effect, while the adverse effects of BPA were not evident. So the vaginal opening data is shown here. On the top, is the continuous dose with both age and body-weighted vaginal opening indicated there were no statistically significant effects. In the stop dose group on the bottom, we had there was a technical error. These animals weren't by protocol design not being weighed daily for dosing at this point, and there was technical error for numerous animals that weren't measured at the time of vaginal opening, as had been requested. So we don't have data for that, but the timing of vaginal opening is not significantly different within the study arm by any BPA treatment. The percentage of abnormal estrus cycles is shown in this slide. The top dose, the top part of the slide is the continuous dose. The bottom is the stop dose. We did, the animals were determined to be, or defined to be, in abnormal cycles by the definitions given in the footnote below: greater than 2 days of consecutive estrus, greater than 4 days of consecutive diestrus, or a very rare event, greater than or equal to 2 days of consecutive proestrus. And those percentages are outlined under each dose group, under the continuous and stop dose. And the bottom line from this is that there was a very pronounced effect of the high dose EE2, but no statistically significant effects on any other dose group in other continuous or stop dose arms. Okay, I discussed earlier the way that we looked at the onset of aberrant cycles, and this is a chart, again, with a continuous dose at the top, the stop dose at the bottom, giving the week and the confidence intervals as well as the p-value. And as expected here for the continuous dose, from what I just showed for the phenylestradiol, the animals were not cycling very early on in the high dose of phenylestradiol group. That was not the case. There was not an effect of the BPA dose groups or the low dose E2. In the stop dose group, we had one statistically significant effect. This was a delay in the time of onset, which would not be an adverse effect relative to the control. One thing to note here is the time, the week of onset here for the stop dose seems to be is quite different from the continuous dose, and is more is out of line, in fact, with the other time frames or estimated times of onset of aberrant cycles shown in the other dose groups. So. For the organ weights, clinical chemistry, hematology, and sperm parameter summaries, again, the summary will be quite short. So the high EE2 dose affected multiple organ weights in the females, while BPA had only minimal effects. And I will show those in the next slide, or a summary of those. The male organ weights were not affected by either BPA or EE2 treatments. Both BPA and EE2 had statistically significant effects on several clinical chemistry and hematology endpoints measured, but the differences were quite shallow, and we did not judge them to be adverse effects. And there were no effects of either BPA or EE2 on the sperm parameters that we looked at. So this is, I'm not showing actual data of the organ weights, is just a summary. There were multiple organs that were affected by the high dose of EE2, adrenal glands, the fat pad, the ovarian parametrial fat pad, heart, kidney, liver, ovary, and pituitary gland. There were few effects on organ weights in the females from BPA groups, and the continuous dose, the lowest dose, 2.5, there was a statistically significant increase of the retroperitoneal fat pad, and a decrease at the high dose of in the continuous, in the stop dose BPA at the high dose on the ovary. And the note at the bottom indicates that neither of these effects were significant when we adjusted for body weight. So there was also an increasing trend noted in the continuous dose BPA liver. In the male organ weights, there were no significant effects of BPA in either study arm. And I just wanted to note that we did not dissect out and weigh the prostate lobes because it was determined that the trimming procedure needed to obtain those weights would have interfered with the processing and sectioning and microscopic analysis, which we considered the more important endpoint for this. In the females, as I mentioned, we had collected clinical chemistry and hematology data, which is tabulated in the report. The effects, there were trends in both the stop dose group, there were trends, and specifically dose groups that were different from controls for several endpoints: mean corpuscular hemoglobin concentration, platelets at the high dose, eosinophils at 250, several other endpoints in the continuous dose. For the EE2, also several endpoints were affected. But as I mentioned in the summary of this, these data points, we did not consider these. Many of them were quite shallow effects and were not considered to be adverse effects. Similarly, in the males, the same sort of thing. We saw several endpoints that were affected with either trends or at specific dose groups in the stop dose and in the continuous dose BPA, as well as for EE2. But again, as I'm not going to show all of the data, but the data are tabulated, and we considered it to be not of adverse nature of these. There were no specific significant effects of BPA or E2 on the sperm parameters that we measured, and I just list the sperm parameters that were measured at the interim sacrifice: testicular sperm counts and the caudate sperm counts, motility, and morphology. So I think I went through that quicker than I had expected to, but this is one of the break points because I'll then go into the histopathology, which may take a little bit more time to explain. But if there's any questions on these endpoints.
Okay, thank you. Any questions from the panel? And again, please, when you ask your question, if you could just identify yourself.
Mike Connor. So the question I have, apparently there were no stopping rules based on survival in this study. So, interesting, because it was done in collaboration with the FDA, who usually do have stopping rules. And some of these groups were below typically where they would say you should stop the study. Anyway, I would say there's a right or wrong answer, but NTP and FDA take quite different approaches to that. So you took the NTP approach, apparently.
Janet Hughes, I had a question about when you said there were some of the, I believe it's the hematology results, that they were, there were significant trends and some groups were significantly different, but that they were not considered adverse events. I'm just wondering what the criteria are for considering something an adverse event.
Well, it's basic. I mean, if it's a large effect, we would it would be considered adverse. But when clinical pathologists look at the data, there's a range of normal for anything, and it was just we didn't have any specific strict criteria, but. In looking at the numbers and some of the trends that the statistics were picking up, we couldn't even really, in the summary table that I've got there in Table 1, I don't even designate a direction because I couldn't even see what the statistical test was seeing there. But anyway, it's based basically on the magnitude of the effect and the opinion of pathologists.
So just this, Dave Dorman, so maybe a clarification or a follow-up to that. So do you have a sense that your clinical pathologists were using normal ranges that had been derived for that strain of rats at NCTR as kind of a historical range to make a determination if it was toxicologically significant?
No, I don't believe that was being used. I mean, there's not that much database at NCTR on those endpoints. But
okay, thank you.
Right.
Okay, if you want to move on to part 2.
Okay. So I'm going to discuss briefly overall the histopathology and then go into the female neoplasms and neoplastic lesions. So the pathology report, which was included as an appendix to the report, was based on the diagnosis of the study pathologist, and then, as Luis described, they were reviewed and agreed to by the pathology working group. And the report itself indicated that many of the lesions identified were common lesions of aging in Sprague-Dawley rats with variable incidences across dose groups. In other words, these reports are completed before any statistics are done, but the pathologist did not signal out anything in their report of particular note that we should be paying our attention to. The lesions represented in the report and the following slides, and I think it's obvious in the report, are selected by the base of the results on the statistical analyses that were obtained subsequent to this. And Louisa had gone over the statistical analysis further earlier. I wanted to go over it briefly again. The CAFE and Poly3 tests were used and considered as the primary statistical test. They consider only incidence for either the neoplastic or the non-neoplastic lesions. The trend tests that are applied are for increasing trend, either positive or negative, with increasing dose. And this is consistent with the general practice of NTP. For the secondary tests that Louisa had described, they take into account severity scores assigned by the pathologist, the Joel-Keir Terpstra, Shirley-Williams, and the relative treatment effect tests. They were also applied for non-neoplastic lesions. So the RTE test was included because it does not presume a monotonic response, as does the JTSW. And again, there are no survival corrections for these tests, which is... So in the subsequent slides, I'm reporting mainly the results of the primary tests. All the results of the secondary tests were included in the tables in the report. And I do note some of these in the slides. All the tests were designed to minimize the false negatives, so they were one-sided. There was no correction for multiple comparisons, despite the fact that we have more dose groups than is typically included in a NTP study. We use the 0.05 significance level at the risk, all this at the risk of increasing false positives. And the final bullet is just statistical significance will not necessarily equivalent, is it not necessarily equivalent to biological significance, and this is not something we came up with for this study. This is the way that the NTP studies and other such studies are always judged in terms of the relevance, and we try to add some of that discussion in the report. So as far as the control incidences, the concurrent controls are always primary. We had continuous and stop dose of animals that were treated differently and were not statistically compared. We did consider comparing those two arms as part of considering the variation in unexposed animals. And similarly, we consider the historical control data as part of the consideration of variation to be expected in unexposed animals. So in the second bullet down here in the draft report, where or end here in slides, where historical control data are listed at the bottom, those are the basis for those are the two NTP technical reports that we conducted several years ago with genistein and phenylestradiol. Those were 2-year dietary exposure studies that were conducted at NCTR in the NCTR Sprague-Dawley rat using the 5K96 diet. Both studies had 2 unexposed control groups. They were a complex design, different from the design of this study, but we followed F1 and F3 generations out to 2 years, and those 2-year animals are what serves as the basis of our historical control. There were no interim data or 1-year data collected in those studies, so we don't have data from those animals. And again, these data are one aspect or one part of considering variability and biological significance. So that's the end of my general histopathology section, and I'm going to move into the female neoplasms. Mammary gland fibroadenomas, as I mentioned when talking about survival, the mammary gland fibroadenomas are a high incidence background lesion in the strain, a major cause of early removals in the females. They were diagnosed in both interim and terminal sacrifice animals with no treatment effect other than a slight decreasing trend in the terminal stop-dose BPA animals that I'll show in the next slides. I just wanted to go over, this is the interim sacrifice animals, fibroadenoma incidence. At the top is the continuous dose, and the bottom is the stop dose. And there are fibroadenomas appearing between the weaning and 1 year, with an incidence varying from, let's say, 8 to 25% across the dose groups. There may have been 1 animal removed or 2 animals removed between weaning and 1 year because of this neoplasm, but it wasn't a prominent issue for the animals. In the terminal sacrifice animals, the diagnoses the percentages are very high, above 80%, between 70 and 80% in most cases. I do note at the bottom that the historical control incidence was 63% with a range of 59 to 69, so the incidences are higher in this experiment for this. There were no treatment-related effects on this lesion, except for the negative trend here in the stop-dose animals. I'm summarizing the data on the female mammary and pituitary glands before I go into the slides showing those data. Adenoma and adenocarcinomas were diagnosed in both the interim and terminal animals, with statistically indicated significance only in the terminal sacrifice animals at the low dose, 2.5 micrograms per kilogram body weight per day in the stop dose arm, and in the high dose of ethinyl estradiol. There were non-neoplastic lesions, which I'm not actually showing, ductal and alveolar dilatation, lobular hyperplasia, which are not related to neoplasms, which were observed only in the high dose. The treatment effect was observed only in the high dose EE2 animals at both the interim and terminal sacrifices. There was a secondary statistical test, the RTE test, indicated an increase in atypical foci in the continuous but not the stop dose BPA animals. And for EE2, but not BPA, there was an increased pituitary adenocarcinoma in the pars distalis, which was diagnosed in the terminal EE2 rats. And this is something that might be expected in this rat, where the estrogen-induced increases in prolactin are correlated with mammary gland neoplasms in the aging animals or in continuous dose estrogen animals. So this is the summary of the mammary gland adenoma, adenocarcinoma combined in the interim or 1 year sacrifice animals. And I will say that I include adenoma, adenocarcinoma in both this and the next slide. Almost all of the tumors observed with adenocarcinomas. There were very few adenomas, but they were combined for the analysis. There were no incidences of these lesions in the stop dose animals at the interim. There were 2 incidences in the BPA groups, lower dose BPA groups in the continuous dose animal, and 2 in the low dose ethinyl estradiol animals in the interim sacrifice. And there were actually statistical analyses aren't carried out if there's not any group with more than 2 animals with a diagnosis. So there was no statistical significance of these. terminal sacrifice animals are a different story. For the top, we have the continuous dose incidence, and then the p-value underneath. At the bottom, the stop dose. In the continuous dose, there was no statistically significant effect of treatment on adenoma, adenocarcinoma combined. There was a significant effect of the high dose phenyl estradiol. In the stop dose group, there is a statistically significant increase at the low dose of BPA of 24% versus 8% in the control. And we discuss in the report compare this to the historical control incidence, which I think I've got here the adenoma-adenocarcinoma ratio. And here you can tell this is the sort of ratio we have. We have here a 1% incidence of adenoma historically, whereas there was a 15% incidence of adenocarcinoma. So I'm just showing the combined on these slides for that reason. But the combined range or incidence was about 15%, with a 12 to 17% instances in the 4 control groups that we looked at historically. So this is above that somewhat, and it is a statistical significant result. Non-neoplastic lesions, as I mentioned, the atypical foci in interim and terminal animals. In the continuous animal, there was a statistically significant increase by the secondary test, the RTE test, that includes severity in the lowest dose BPA group, both at the interim and terminal sacrifice levels. There was no statistically significant effect in the stop dose animals of either the interim or the terminal sacrifices. And I did note a historical control range down here of 11 to 37%, so quite a broad range in our 4 control groups used previously. The pituitary, I've got a footnote here that actually this combination was not included in the statistical analysis in the report. The data are there in the pathology report, but not in the statistical analysis. Combined adenoma carcinoma in the pituitary. There was a statistically significant increase in the high-dose phenyl estradiol animals. No significant increase in the BPA treatment groups of the continuous or stop-dose groups. So in the report, and some of the couple of comments dealt with the mammary gland neoplasm at the low dose. And here I've listed some factors that we use in questioning the biological significance of the low dose BPA statistical result. First, it occurred only in a single dose group. It was not dose responsive. So there was no indication of any sort of trend to our vision in those results. It occurred only in the stop dose arm. It did not not occur in the continuous dose arm. And I guess that you could formulate hypotheses as to how that would occur, but it was a surprising finding in our view. The incidences were variable across all dose groups in continuous and stop dose arms. So that first slide where I showed the incidences, the range of incidences across all the dose groups was approximately equivalent in the continuous and stop dose range. I think there was a 20% incidence up in one of the intermediate BPA dose ranges in the continuous dose group, and then the 24% incidence in the low dose of the BPA exposure arm. So the statistically significant incidence was near the high end of the control incidences observed in the previous NCTR studies, and the control incidences were lower than the observed in previous studies, and were also the controls in the stop dose were the lowest in the other 2 control groups in this study. And finally, this is not really associated with the BPA treatment neoplasm, but when I put this in to indicate that the EE2 increase in adenomas, adenocarcinomas was more interpretable to us The reference high dose showed of ethyl estradiol showed a pattern consistent with estrogen-induced mammary neoplasms in Sprague Dawley rat, where both the mammary adenoma, adenocarcinoma, and pituitary neoplasms in terminal animals were observed, or dose increase of those tumors. There were other lesions in the female that were diagnosed in the uterus. There were both neoplasms and non-neoplastic lesions. Actually, uterine stromal polyps were the only other neoplasm in females in which there was a statistically significant effect observed, and that was a significant trend in the interim sacrifice continuous dose animals. There was a significant negative trend and reduction in the high-dose BPA group in the terminal sacrifice stop-dose animals. And there was a significant increase as far as non-neoplastic lesions in apoptosis in the luminal epithelium in the interim sacrifice females in the continuous high BPA and the high-dose ethyl estradiol groups. These are the interim sacrifice stromal polyps in the uterus. As I mentioned, in the continuous group, there was a statistically significant trend in which there was a 4% incidence here in the vehicle control group. In the two highest dose groups of BPA, we had a 15 and 12% incidences. There was not significant increases in the EE2 groups, and there was no significant increases in the stop dose BPA group. And I've got this footnote here. I should have mentioned this at the first show of the any histology slide, but I hope people are interpreting this correctly. That's p-value that's under the vehicle group is the trend for the BPA dose groups. I have the EE2 groups attached on here for comparison, but they're not involved in that trend test. So sorry. In terminal sacrifice animals, there was not observed increase in stromal polyps in the uterus in the continuous dose group for either the BPA or EE2. There was not an increase in the stop dose group. There was a negative trend, and the highest dose group was significantly lower than the control group for the stop dose animals. In terms of non-neoplastic effects, we did this diagnosis, luminal epithelium apoptosis, is something that would happen normally during the estrous cycle. The pathologist notes it when it's higher than normally expected. There was a significant trend in the continuous dose BPA animals and a significantly difference from control in the highest dose of BPA, 25,000 micrograms per kilogram body weight BPA in the continuous dose animals, as well as in the high dose EE2 animals, a significant different comparison from control. In the stop dose animals, there were no significant effects. In the ovary and vagina, there were no neoplasms. There were treatment with any statistically significant treatment effect. However, there were some non-neoplastic lesions in both the interim and the terminal sacrifice animals. The incidence of cystic follicles was increased in the stop dose interim, high dose BPA females, and there was a dose trend that was evident. There was high dose EE2, which was a continuous dose only, of course, also increased the incidence of cystic follicles. There was epithelial hyperplasia diagnosed in the vagina in the continuous interim high-dose BPA females, and there was a dose trend evident. Incidence was also increased in interim high-dose females. High-dose EE2 females, excuse me. And epithelial hyperplasia in the vagina was elevated, statistically elevated above controls in multiple BPA dose groups in the continuous dose terminal females. So this is the cystic follicles in the ovary. Continuous dose, the high dose of EE2 is statistically, well, it's 100% animals versus 35% in the vehicle control. In the stop-dose BPA animals, we had a 82% incidence versus 25% in the controls. And there was an apparent dose trend evident here, and there is an elevation here at the highest dose groups. In the vagina, non-neoplastic lesions here, this slide indicates both the interim continuous at the top, continuous and stop dose, and then at the bottom, the terminal continuous and stop dose. And there was a significant effect, a trend, and also the high dose versus the controls for the highest dose of BPA, 25,000, and for the high dose of EE2, 0.5 micrograms per kilogram body weight per day. In the terminal continuous dose animals, where there's not much, the ovary is basically not functioning at this terminal phase. But there is some elevation of hyperplasia in the epithelium of the vagina at multiple dose groups, and this one dose group in the middle was indicated only by the RTE, not by the primary tests, but there is an elevation versus the control here, not evident in the high dose of ethinyl estradiol. No observed effects on this endpoint in the stop dose animals, either in the interim or the terminal sacrifice phase. So the next slide is going to include a summary chart of all the significant effects. In the report, I have a lengthy table one. We have a lengthy table one, which extends over multiple pages and lists every endpoint that was measured and whether there was any statistically significant effects seen in any dose group. For this slide, I'll show in the next slide, I've removed anything that didn't show any effects in any treatment group in order to make it able to be fit on a slide, for one thing. But the full table is certainly in the study report. The high dose of EE2 is probably evident from the slides I've shown, will certainly be evident from the next slide. Induced multiple effects, particularly in the female reproductive organs, including increases in mammary gland and pituitary neoplasms. Statistically significant effects of BPA were observed in multiple tissues, various dose groups, including the lowest dose. Our interpretation was that there wasn't a coherent pattern of effects. And the slide is shown here. I've got the stop dose BPA. I've broken them into endpoint groupings, and then I list specific endpoints here, which are unlikely to be able to be discerned, but I've showed most of those on the slides previously. Stop dose BPA from 2.5 to 25,000 summarized here. Continuous dose BPA here, and then the two EE2 groups. So the striking feature of this is the multiple dark bars over on the high dose of E2, where there were multiple significant effects in multiple tissues, both with histopathology and with non-histopathology endpoints. Within the BPA groups, there are significant effects that we discussed throughout the endpoints measured, particularly in the histopathology endpoints, and at multiple dose groups in both the stop and the continuous dose BPA. I split these out and we did set 0.05 as the significance level in this study. Sometimes, or often in reviewing cancer studies, or people use a higher stringency p-value for common tumors versus rare tumors, and I've just split these out in terms of the significance levels in this dose chart. And most of the highly significant findings were, of course, in the potent estrogen group that we tested. Either in these slides, I did show some of the results of secondary tests. I'm not going to show a similar slide using the secondary tests. In most cases, were the non-neoplastic lesions that were analyzed by these secondary tests. If the CAFE or poly3 were significant, the secondary tests were also significant. There were cases in which there were additional statistically significant results indicated, and these are all presented in the report. And we felt that there was not a clear, it was similar to what I just showed for the BPA groups, and our evaluation was that there wasn't a consistent pattern of effects that were observed. In the few cases, there were multiple contiguous groups that were indicated as significant, and the pituitary hyperplasia and the cardiomyopathy I've shown on the next two slides as examples of these. Common features are relatively high background incidences, and what we interpreted as minimal percentage differences above the background. So these are the pituitary values. At the top, I've got, again, this is the interim continuous dose and stop dose, and then below the terminal continuous dose and stop dose. There was high incidence of hyperplasia in the controls here, 78%, 90%. No difference in the stop dose group. There was an elevation at the high dose in the interim animals at the 25,000 group, and in the EE2, where virtually all the animals had hyperplasia. In the terminal groups and the continuous dose, there were no statistically significant effects. In the EE2 groups, no significant positive effects. In the ethinylestradiol groups, in the terminal stop groups, there was an increase in the two lowest doses of BPA indicated by the relative treatment effect test, 64 versus 51%, 71% versus 51%, and elevation 3.3 versus 2.8, 3.1 versus 2.8 in terms of severity scores, which were similar across the board for these. Cardiomyopathy was the other endpoint that I had indicated, where there was the secondary tests, but not the primary tests, gave multiple contiguous doses that were significant. There was nothing significant seen in the interim 1-year animals, neither continuous or stop dose animals. In the terminal animals, the incidence had, the only significant increase here was in the high dose E2. There was a negative finding in the 225. In the terminal stop dose animals, there were multiple dose groups, all except, and I can't quite understand this, the fact that this lacked significance in the statistical test, except perhaps it has to do with the N value that was used. I just don't understand that one. But anyway, across all the dose groups, there was the RTE test indicated an increase in cardiomyopathy, which is a prevalent lesion in the older females. And that was all of the female histopathology that I was going to show. And if there are any follow-up questions at this point.
Okay, thank you. I'll turn it over to the panel. Dr. Rosal.
Tom Rosal, I just had a question. I wonder if you could refresh us on the pharmacokinetics of EE2 in the chronic study.
We, Dan Dorgey's laboratory, did pharmacokinetics of both the BPA and EE2 in conjunction with our 90-day study, in which he evaluated, similar to what we did with BPA, the dose dosed the animals with EE2 at various ages, and I believe it was PND4, PND21, PND80, and then measured the levels in the serum at approximately the time of Cmax. So, and in that study, he found that the levels, similar to what happened with BPA, levels are very high at the PND4, lower at 21, and lower still at PND 80. So I can't give you the actual values, but it's known. I know it's known. It's certainly in the rat versus a human. The oral bioavailability of E2 is much lower, but that almost all that work is done in adults. I can't compare that in the neonates, but.
I thought there was some reference in the report to blood levels at 1 and 2 years from previous studies. Did I misread that? all that data was just from the 90-day study?
We did not collect data from 2 years. Final. Yes. Mhm.
Questions? Okay. Go ahead.
Janet Hughes, I had a question about, I'm just, because the p-values are based on the poly 3 test, and the poly 3 also gives you an incidence. I'm curious why you report the simple incidence when the test is based on the poly K incidence.
I think that has mostly it's commonly done in NTP reports. I did in some of the instances in the study report when I was first drafting it that I did include, and I think I still have in some tables, the poly 3 incidence, which many of the reviewers asked me to take out. But I think you're right that is what the comparison is based on, and it makes more sense. So, but we did go back and look at poly 3 incidences and the historical controls and try to compare them with this. But they were also, the poly 3 incidences were higher than the simple incidences in that point. So I understand your point in terms of understanding what the test is actually seeing.
Okay, thank you. Part 3?
Part 3. Okay, so now I'm going to go on to discuss the male. And this is, as I've been doing throughout, I provide first a summary slide of what I'm going to talk about. There were no statistically significant increases in any organ-specific neoplasm in the interim or terminal sacrifice males in the continuous or stop dose arms for either of BPA or E2. There was a significant increasing trend in the stop dose BPA males for systemic malignant lymphoma, which was diagnosed in several organs: liver, dorsolateral prostate, bone marrow, spleen, and kidney. And the vehicle control incidence there ranged from 0 to 2%. The highest incidences were the 25,000 BPA per kilogram body weight per day dose group, and ranged from 9 to 11%. 2 neoplasms were diagnosed in the dorsolateral prostate lobes: an adenoma at 25,000 micrograms per kilogram body weight per day, a BPA, and an adenocarcinoma in a 25 microgram per kilogram stop dose BPA animal. And of course, these weren't included in the statistical analyses due to the low prevalence of those tumors. Adenomas were diagnosed in the ventral prostate across all dose groups with no statistically significant differences from controls, so I'm not presenting those data. In terms of non-neoplastic lesions, there were statistically significant differences in the males observed in the epididymis of interim sacrifice animals, and in the pituitary and dorsolateral prostate and mammary glands of terminal sacrifice animals. And I'm not showing data for the ventral prostate, but I just wanted to note, make a note that in the ventral prostate, the suppurative inflammation and fibrosis were decreased relative to the controls. I didn't show those data. That wasn't, our aim was not to see what was decreasing, but if you do look through statistical tables, there are a lot of effects that are negative, indicated as negative relative to the control across the study, the endpoints measured in the histopathology analysis. For the non-neoplastic lesions in the epididymis, in the continuous and stop dose arms, here at the top, well, I've got, let's see. Yeah, the exfoliated germ cells are shown at the top. At the bottom is lymphocyte infiltration in the continuous dose first, and then in the stop dose. For the continuous dose animals, there was an increase in exfoliated germ cells seen in the highest dose of BPA. There was also an increase in lymphocyte infiltration seen at the highest dose of BPA. And by the RTE test in the high dose of E2. I will note down here that there were no treatment differences in any testicular lesions to be associated with these epididymal findings. at the lowest dose of BPA in the stop dose group, there was an increase incidence of exfoliated germ cells in the stop dose group. In the male pituitary, there was an increase incidence of hyperplasia in the pars distalis at the highest dose group of bisphenol A, 25,000 micrograms per kilogram body weight per day, in both the continuous and the stop dose arms, and also with the highest dose of the E2 in the continuous dose arm. I did provide a historical control incidence down here, 26% with a range of 16 to 39%. So this is about in the range, the median range, but the highest ranges encompass what we've seen in the past for this endpoint in this animal. In the dorsolateral prostate, this is a high incidence lesion, both in the interim sacrifice, here at the top, continuous and stop dose interim, at the bottom, continuous and stop dose terminal sacrifices. There was, by the RTE tests, indicated a significant increase in several BPA dose groups. And by the poly-3 test, in the lowest dose of BPA at the terminal sacrifice continuous animals. I note at the bottom both the historical control range, which was lower than we see here in the background, and we think this might be related to the fact that in the past studies we were only taking one section of the prostate, whereas as Louisa had described in the study description in this, because of our interest in the dorsolateral prostate in this particular study, we had the pathologist examined step sections through that lobe of the prostate. The mammary gland non-neoplastic lesions, I put this slide in here first because there was by one of the primary tests, an increase in dilatation of the alveolus noted at 2.5. The pathologists indicated they did not consider this a pathologically meaningful lesion, but we were interested in both treatment groups of seeing any effects in the male mammary. In the 90-day study, this bottom bullet here in the footnote, in the previous 90-day study with BPA and E2, we had observed ductal and alveolar hyperplasia in the animals. The highest was in that study, the lowest dose of BPA, 0.5 micrograms of E2 per kilogram body weight per day. And earlier in the dietary E2 studies, which were much different exposure, we had noted induction of male mammary gland hyperplasia at 2 parts per billion, or approximately 0.2 micrograms per kilogram body weight per day in younger animals at PND 140, but only at higher doses, 5 and 25-fold higher in the 2-year males. So we think we did not observe any ductal or alveolar hyperplasia in the male mammary glands, and we think that this is due to the age of the animals at which we were examining in terms of the E2 effect. So I have a similar summary chart for the significant effects of BPA and EE2 in the males. In contrast to what you saw in the females, the effects of EE2 were relatively minimal. As with the females, there were statistically significant effects of the BPA in multiple tissues and various dose groups. But again, to us, it was hard to discern a generally interpretable pattern. There was an increased incidence, as I mentioned, of hyperplasia in the pars distalis and the pituitary at the highest BPA dose groups in both continuous and stop dose arms. And the effects in the epididymis were observed in the high dose of BPA without findings in the testis. And here, again, I'm not going to go through this in detail. It's just a visual, for visual effect mostly. But again, there are effects at EE2 that are not as strong or as prevalent as in the females. There are multiple effects, most of which I covered in the previous slides, in males at various dose groups, in both the stop and continuous dose groups. And when we say in the report that we make a mention at some point of not seeing correspondence between the stop and continuous dose, we mean that we're seeing some lesions are appearing in the stop dose, some lesions are appearing in the continuous dose, and either within those dose arms or across those dose arms, we can't see a consistent pattern of effect. Again, I'm not showing a chart of the effects of secondary tests. Again, as with the females, in most cases where the primary test showed significance, the secondary test also showed significance. In some cases, there were significant results with the distribution across dose groups and study arms that I just mentioned that were not clearly interpretable to us. In a few cases, as with the females, there were multiple contiguous dose groups indicated as significant. And I'm not going to show again, I had shown earlier that example of suppurative inflammation in the dorsolateral prostate, where there was a relatively high background, and we consider to be minimal differences from controls across those affected groups. And I've got to stop here, and I have one summary slide after this, but for everything, so that.
Well, so why don't you go ahead and show that slide?
Well,
just if you.
Okay, so this was a summary, and this is an overall summary taken from what was the verbiage in the report. So we interpreted the data as saying that BPA had effects that were certainly distinguishable statistically from background, but with questionable biological relevance. The effects were largely not dose responsive, often occurred in only a single BPA dose group, and not a clear pattern of consistent responses within the stop dose, continuous arms, or across those arms. The observed variation in endpoints, particularly below the highest BPA dose, we felt were within the range of normal biological variation in this strain of rat. Some of the effects observed at the highest BPA dose may be treatment related, and I've listed some effects in the females and males where we thought that might be the case. And the EE2, particularly the high dose, produced clear adverse effects in the females. And that's, the slides after this, I think we're going to hold off on until later in the day. Is that right, Mary?
Right.
Yeah, okay.
Right. Okay, thank you. Any clarifying questions from the committee or Okay.
Must have been a lucid description.
So I have one question, I guess, and it relates to Dr. Walker's presentation. He said that this was designed not to be really a hazard ID study per se, but was really trying to do a dose characterization or look at dose responsiveness. So a priori, had there actually been any hypotheses with respect to what the dose response, the nature of the curve, monotonic, non-monotonic, may have actually existed? So was there, since that's the goal of the study, right, was there any hypothesis with respect to that?
There was not a statistical analysis designed in to look for curve fitting or that sort of thing to look for a non-monotonic dose responses. The only thing that we had done was when we were doing the individual comparisons against the control to see if there were any statistically significant effects. But no, we did not. There was not, for this study, formulated a hypothesis for testing that.
Nigel Walker. To the extent that we strongly felt that we wanted to cover a wide range of doses, the hypothesis was we did not expect, the null would be, we expect nothing down the low end, so we'll just focus on the high end, which is the traditional way of doing things. We definitely went against that. I mean, that was a conscious decision that we want to go from essentially more of a margin of exposure based. So as Louisa pointed out, it was around tenfold above where we figured we might have background rate, and 25,000 was, everyone agreed that was about the top end. What we would, to go even further was people didn't feel that there was a lot of value when we were discussing the final design amongst the whole steering committee and the grantees. We felt like this would be balanced across the dose response range. So the hypothesis was almost it could happen anyway. So therefore be balanced, not going to focus on one particular area. And that's actually a challenge when you're doing dose response studies. When we've done past chronics where we've done dose response studies and we've used modeling to identify a specific range where we were expecting, Furan, for example, was a 2-year study we did, where there's specific modeling to identify the specific range and then focusing in here. But in this one, we had no a priori expectation. We just knew that reported effects had occurred anywhere from 2.5 to 25,000 and above.
Okay, thank you. Any other questions? Okay, well, thank you. Sure, just use your microphone, please.
Luisa Camacho. Just to address the question about the internal dosimetry of EE2. So we did not measure it at 1 year and 2 year animals, but based on our 90-day study data, we could be sure that the levels are very low. So in the 90-day study data, we collected internal dosimetry for EE2 at PND 4, 21 and 90. And by 90 days of age, the levels were below the limit of detection. So they expect the same to happen at even older animals where the metabolic capacity was fully developed.
Okay, so we're now in the agenda of trying to acknowledge the public comments. And so there should be a slide showing those organizations that provided written comments and individuals that did. So for me, that's a little hard to read, but I'm not going to try to read all of them. But I think what I will state is we received a wide range of public comments. And I appreciate the people that provided public comments. They were actually quite thoughtful. And I think the peer reviewers on the panel had an opportunity to consider them as they were drafting their comments in today's discussion.
Is there a page 2 or is this just one? Okay, thank you.
So do you want me to read them into the public record or just there it's available, right? So
Yeah, and the URL is there, or they're available off of our main meeting page that you can, and we provide the URL also, and they were provided to the panel.
And I'll make an aside comment, which I actually found interesting, and just because it's my prerogative, I guess, because I got the gavel, so I can do what I want. What I also found interesting was several trainees provided comments, and I actually thought that was something worth noting today because I think a lot of times advisors of graduate programs don't point out that opportunity to trainees. So I'll actually give a shout out to those couple of students that provided comments because that's a pretty bold thing to do, and I'll just give a shout out to those students that did that. So, okay, so we're going to switch now to oral comments. And so let me just have a sidebar here with Dr. Wolf. Do we know if the person from, or both are on the phone, or we could double check?
line is open. Hi, I'm hoping you can hear me.
We can, so
Great, thank you. Thank you for the opportunity to provide comments to you. I am going to keep this brief because I was allotted 5 minutes and focus really on just a handful of points that I think some were raised in written comments and others perhaps were already raised this morning on the webinar. Next slide, please. So I'd like to start by pointing out some perhaps concerns or more interest in the framing of the study. In 2013, a paper was published by authors from National Toxicology Program, NIEHS, and the U.S. FDA, which helped shape my understanding of what the goal of the CLARITY project was, both the core study and the accompanying academic portion. And I think it's important to point out that CLARITY-BPA was really identified as an opportunity for filling knowledge gaps, identifying ways to improve or inform chemical risk assessments, and identify new methods or endpoints for regulatory hazard assessments. So I think that part of what is missing, not just from this document, but in the way that this document was discussed in public forum, including in interviews with NPR, is a failure to acknowledge that this core study is really a part of a wider discussion about how to identify new methods and new endpoints for regulatory hazard assessments. I think part of what I've learned from listening this morning is that it's now less clear to me what the role of NTP versus NIEHS versus US FDA and the NCTR lab was in each step. So it's less clear to me who ran the study and who was responsible for analyzing the data. It's also unclear from the actual report that this was supposed to be a dose-response study. And so I think that that should be made clearer in the report. And I'm also confused because if it is a dose-response study, it did not include the dose that's been widely identified previously as the NOEL dose, which is 50 milligrams per kilogram per day. Next slide. I think that there's a lot of positive things about the conclusion of CLARITY-BPA, one of which was that this offered the opportunity for academics to participate in a project and utilize tissues from GLP-compliant laboratories. But the failure to acknowledge the role of the academics, I think, is really a shortcoming of the report. Next slide, please. I think that there's some really interesting data here in the report, including endpoints that were not analyzed, and that also leads to my concern about who's responsible for analyzing the data. For example, I'm showing you data that I graphed based on the report that illustrates the percentage of females that had success breeding. And this is data that was taken directly from the report, and then a simple chi-square test suggests significant differences in breeding success at the 3 lowest doses of BPA. So I suspect there are other endpoints that are sort of hidden throughout the report that hopefully additional analyses can be conducted and will include experts in biostatistics, which I am not. Next slide, please. I found it very interesting that there were numerous effects observed at low doses, particularly because these effects observed at low doses overlap with outcomes that have been reported in some cases, dozens of academic studies. Of course, academic studies usually use those endpoints as a launching pad to evaluate mechanistic relationships, mode of action relationships, and that isn't the point here of the core study. But I think the pattern here is really interesting. A few questions that were raised. One, are the pathologists that perform these analyses blind to treatment group? And was it decided a priori which effects were adverse and which were not? Because it seemed from this morning's report that some of the data interpretation ignored statistical significance and instead focused on opinions from pathologists, which may introduce bias. Next slide, please. I think that there's a really important possibility of stress, and so I've graphed here just the differences in body weight for the 2 control groups over time, suggesting that perhaps the handling of animals has changed the biology of these animals. I would be interested in seeing the prolactin results that were cited this morning. I can't find them in the report. And I would note that the comment that estrogen-driven prolactin increases are correlated with neoplasms I don't know of any citation that would support that. I'd also be interested in seeing corticosterone results and also potentially histopathological analyses for the esophagus to determine whether there was damage being done by gavage. There were also differences, I'm just showing you differences that I noted for body weight between the control groups, but there were also differences in control groups for other endpoints, like the timing of the onset of aberrant estrous cycles. Next slide, please. I would note that when you actually look at body weight endpoints, the differences between the stop dose group and the continuous dose group suggest, again, the potential that stress could be interfering with the ability to detect differences between these groups. Next slide, please. One of the things that I was really looking forward to in this report was based on what was again reported by Shug et al., authors from NTP, NIEHS, and US FDA, that the core study would provide information about internal dosimetry, also quantification of background of BPA and other sources of potential endocrine activity. We know that there was a problem in the pilot study with background exposure to BPA, as well as the potential contamination with BPA during serum collection. So I think it's the lack of exposure data is a serious concern here. Next slide, please. Finally, I'm concerned about the proposed use of historical controls and potentially the use of those historical controls to ignore or to try to downplay the effects of significant effects of BPA on adenocarcinoma or adenoma in the female mammary gland. I'm particularly concerned because this webinar identified that the historical control data came from NTP studies 545 and 548, one of which was published in 2008 and another published in 2010. It's generally agreed that historical control data should come from a period of 2 to 7 years, unless there's evidence that the endpoint is stable over longer periods. So I'm interested if there is such evidence. Further, both of these studies used non-gavage methods of administering the compounds of interest and used polycarbonate cages, suggesting, next slide please, that there are potential differences between the concurrent control groups used in this study and historical control groups. I'd also urge the release of historical control data for all of the endpoints so that they can be compared for other endpoints, not just for the ones that were statistically significant, because perhaps there are things that are significant but have been missed. And of course, it would be important to know about background contamination of the chemical in question, but the use of polycarbonate cages in those historical control studies suggests that they are not appropriate for use. I'll conclude there and happily take any questions you might have.
Okay, so we'll have time for one or two clarifying questions from the panel. Okay, I do not see any. Thank you. So is our second...
You hear me?
We can hear you.
All right. So Cécile Michel for the French Agency for Food, Environmental and Occupational Health and Safety, France. As you might know, ANSES is having an interest in BPA as we are responsible for many of the regulatory actions taken in EU on BPA as a chemical substance under REACH. So we would like to congratulate NTP for the work performed and thank you to give us the opportunity to discuss the major issue of the study. As the time allocated to me is short, I will focus on the point that bother and question us. So to start with, we have some question regarding information missing. And I was not sure, maybe it was specified this morning, but as we are not sure on the hormonal status of the animals and in particular the œstral stage at which female, especially at one old of age, their stages, we couldn't find this information in the report. So we think it's an important parameter to be specified. Having heard carefully your presentation this morning, we also wonder how it was technically performed that the pups were exposed and what technique was exactly used. We have some difficulties to understand and to visualize exactly the performance of this exposure. We wonder if we understood correctly that they were exposed, the pups were exposed by gavage. Okay, so now regarding other points that you received by written, we have questions regarding exposure. We have two questions regarding exposure. The first is concern the F0 breeders. As we understand that their housing conditions may have led to exposure through BPA, and that BPA was not controlled, so we would like to have a confirmation on that. We also have a question regarding the difference of exposure of the groups that were exposed through systemic. There were two different formulations, and we wonder if it affects the systemic bioavailability. We have one big non-understanding, really, regarding the statistics that you performed. We understand the rationale why you had to split the high number of animals into different cohorts, but from our understanding, the five kind of study performed could be considered as independent studies, and we wonder if you have tested anyhow how these groups could be considered as independent. We would like to ensure that there were no effects on seasonal effects or technician assigned, and we thought that this could be considered as a parameter within your statistic. Now, regarding the results, we sent you some questions. So regarding the results with the positive control, we were quite surprised that some of the parameters that we normally expect to be responsive to ethylene turned out to be negative, and we are willing to have some discussion about these results and why these results were not positive. We are talking especially on vaginal opening timing, sperm parameters, and mammary gland in male. Sorry. Regarding the control group, there were also 2 points that draw our attention on. There were some non-neoplastic lesions that were observed with high incidence in animals continuously exposed to vehicle, in particular endometrium cystic hyperplasia. And we would welcome details and discussion on this peculiarity. Another peculiarity was observed within the female reproductive system, for which the two negative control groups are not homogeneous. So it's one of the comments that were performed before by Laura. So we would be happy to hear some explanations or discussions on that point. Now, regarding the results on BPA itself.
So, excuse me, if you could try to wrap your comments up in 1 minute or two, that would be great. Thank you.
Pardon me?
Could you try to wrap up your comments in the next minute or so? Thank you. Yeah. We wonder if we have some. There were some effects on the mammary gland, and we would be happy to see that confirm those that were found in the preliminary study.
So we would be happy to have to be able to see some discussion on that. So we are looking forward the grantee studies and more detailed discussions and comparison with the existing literature of the core study, in particular on the peculiarities and the resistance BPA that we provided to you by written. And we we all agree that we did not expect any undiscutable adverse effect with this CLARITY study, but rather subtle changes. And I think it's important that this discussion is available on the core data. And we would like to conclude to say that the data you provide confirm the work we have performed previously, and we thank you for that.
Okay, thank you. Any clarifying questions from the panel? Okay, there are none. Thanks again for your comments. Traditionally, we can open up the floor for very brief comments from anyone who's here. Gone. Okay, so we are approximately 20 or so minutes ahead of schedule. So what we're going to be doing is breaking for lunch right now. And rather than reconvening at 1 o'clock, I'd like to reconvene at 12:45. That actually gives us a smidgen more lunch break, but we'll be back here in this room at 12:45. Thank you.